Welcome to 17 Minutes of Science. My name is Sarah Cheeseman, and I am a technical solutions scientist at In Vivo Biosystems. And it is my real pleasure to welcome our guest today, Dr. Rita Fior, who is a researcher at the Champalimaud Center for the Unknown in Lisbon, Portugal, which is just such a fantastic name for a research center. So today we're going to be talking about developing novel assays in zebrafish for therapeutic screening in cancer. So Rita is a developmental biologist who began using the zebrafish model when she was a postdoc. And now as an independent researcher, she's interested in using fish to understand cancer, especially through the lens of personalized medicine. So in her words, off her bio on her wonderful website, which I encourage you to visit, is cancer is as unique as the person fighting it. And this is why we need to have a personalized approach to cancer treatment. So I'm going to welcome Rita, let her say a few words, and then we'll turn over to the questions. And I'm starting the timer because I always forget to do that, but we're on. So Rita, welcome.
Thank you. Thank you so much for the invitation. Thank you.
Well, we're glad to have you, and we appreciate that it's the end of your day in Lisbon. And so she's making time for us at the end of her busy day. So let's start with, why don't you tell us about how you landed on using zebrafish as a model to approach these personalized medicine questions? I think people sort of naturally gravitate towards thinking about mammals. So if you could tell us about the fish as your choice.
Okay, so basically I did my postdoc in zebrafish, studying segmentation. So the formation of the somites, the rhythmic formation of somites, and how do you make somites in zebrafish. But, and then I switched to cancer, and really I switched to cancer. Well, I think many people have this experience, a personal experience, where I realized that there were several options, equivalent options for many patients. So the patient goes to the oncologist and is diagnosed with a type of cancer, stage of cancer, but then in the guidelines you might have, you know, option A and option B, which are equivalent, drug A, drug B, and there is no test like an antibiogram for bacterial infections for chemotherapy. And that was really, for me, very stressful. It was a shock, really. This started with a conversation with a friend, oncologist, that I was discussing the chemotherapy that my mother was taking. And then she said, well, in my hospital, we don't give that, we give another drug. And I was like, what? Sorry. So I was like, oh my God, if this doesn't work, but if my mother went to another hospital, it might work. What is happening here? I really did not know how cancer treatments were, the guidelines were made, actually. And then I realized that of course, you can have equivalent options. It was nothing wrong with that. It was just, they were just equivalent in different hospitals, and just doctor chose one over the other. But I was like, oh my, well, but maybe this would not be the first choice. And what happens is patients, if the drug A doesn't work in the first place, then you'd switch to B. But if you knew in advance that B was better, you would avoid all the side effects and all the toxicity, and you would don't lose so much time.
And gain time, hopefully.
Time. And so I think I'm not being very clear, but this was really my switch to cancer. And why zebrafish? Because I did my postdoc in zebrafish. And also, so I tried to understand if there was a test. There was no test, nothing in vitro. In vitro was not working, so there was no predictive value of between in vitro and the patient. So the gold standard at the time was mouse xenografts, of course, and they've been a great model to study cancer and also for drug prediction and to do the mouse avatars. So this was really the gold standard in the field when I started. But why did I change? I wanted to invest in zebrafish is because of time, because mouse xenografts take months to implement, or at least 1 month. So it takes a lot of time to grow the sample that comes from the patient. And then you have to increase the cohort of animals that have the tumor to then have a group of animals to test and a group of animals of control. So it takes months. So I wanted a fast system. And then I was in Woods Hole doing the Zebrafish course, and we had the screening module with Randy Peterson. I was like, why don't we try this in Zebrafish? It was a bit like this. Of course, there were already people trying it. I wasn't the first. Then when I went to the literature, of course, there were, so I was not the pioneer. There are other people that started the model. And so I thought we should try to make it a reality for patients.
That is an interesting story. It makes a lot of sense about the timeline, and that's a lot of reason people turn to Zebrafish, right? Can you tell those of us who are watching who are unfamiliar with a xenograft what that is? I also love that you use the language of living test tubes, so maybe you could type it together for us.
Okay, sorry. So what is a xenograft? So in this case, the zebrafish, it's the host. So the idea is to take a sample from a tumor from a patient and then test these cells from the patient directly. But the idea is that you put these cells inside a living animal, which can be a mouse, or in this case, the zebrafish. So this is a xenograft. This is what we called a xenograft. So zebrafish xenograft is when you put tumor cells, human tumor cells inside the fish, or a mouse xenograft or avatar. We now call it avatar also because it's like having a bit of the patient inside the animal. But the idea is to test the cells before you give the therapy to the patient. So this is the big goal, that we can test before giving to patients which is the best therapy, at least the therapy that you'll have a higher probability of a good response. Doesn't mean it will be 100%, but the way we are doing is still doesn't make sense. If we think we go to Mars or to the moon, or we fly planes every day, we have amazing technology, for everything. I think it's amazing. We are talking to the US, and I think internet is amazing. But to study biology, we really have amazing technology. We can sequence genomes every day and so on. But if you're a cancer patient, you still don't have a test. So
right.
This is
so trying to make those linkages that you're doing of bringing these things together. It's powerful to hear you talk about avatars. We like that word too at In Vivo Systems, so we use it as well. When you're doing these experiments, I'd love to hear a little more about, I know you're interested in breast cancer. We're also going to touch briefly on your colorectal cancer work. Where are you putting the cells in the fish, and at what stage are you doing that?
Okay, so we inject 2 days post-fertilization embryos, zebrafish embryos, so they're very little. And then we just next day, so after injection, so we inject in the perivitelline space. I don't know if people know, but it's near the heart, a bit down. We try to inject away from the heart, but it's in more or less the same space. But we have to avoid the heart so that you don't create cardiac edema. And then next day, what we do is we screen our fish and see the well-injected fish continue the experiment, and the ones that are not well-injected are simply discarded. And then so this day we start the treatments, and then 3 days after we stop and sacrifice the xenografts, and then analyze by confocal microscopy. So what we then want to see is if the drug induces cell death. So we want to see which is the drug that actually kills the cells, right? And you reduce the tumor size and you induce apoptosis. This is our readouts generally.
Do you see around the site of the xenograft remodeling in the host in accommodating that foreign tissue? I know this intersects really interestingly with the innate immune response as opposed to adaptive immunity. We may be too deep to go into that right now, but I understand that's a big advantage of why you're using the larva.
Yeah, exactly. And we just published very recently a paper on that. So yeah, what we can see, which is, I think it's absolutely amazing, is that depending on the tumor cell or the types of tumor or the tumor cell, it's not even the type, if it's breast cancer or colorectal cancer, each cell is different from the other, is that each tumor can generate a different tumor microenvironment. So you have recruitment of macrophage, neutrophils, at least that we know, of blood vessels if these cells are highly angiogenic, and we can see this in a very quick time frame. So in 2 days we can actually see vessels coming in, or a macrophage, well, 3 hours they're coming in, and neutrophils. So and what is really amazing is that each tumor is able to recruit a different tumor microenvironment.
That is interesting. And I guess with the beauty of the fish, you're able to visualize all that because they're so clear and accessible, which I suppose is an advantage over too, isn't it?
And also transgenics. So we can use the transgenics to label the cells and actually, do movies. And
yeah, you must make beautiful pictures. Well, judging from your website and what's behind you, you do take lots of pictures. For those interested, she's got a phalloidin stain in her backdrop. Let's touch on the paper. So congratulations on your Nature Communications paper this week that you just referenced. We were excited to see that come across all of our zebrafish alerts describing the colorectal xenograft assay. Can you tell us how that work is arcing back towards serving patients?
Well, this one is a bit maybe far away, but not so far. So, yeah, what we saw was this part of the tumor microenvironment. So it looks like, so what we are proposing in the end of the paper is that just like zebrafish can be a reporter for angiogenic potential. So when you inject tumor cells that express a lot of VEGF, because everything is so conserved, endothelial cells come in and form the vasculature surrounding the tumor. So actually you put, if you put tumor cells that are highly angiogenic, you see a lot of vessels coming in. If they're not, you don't see. So it's like a reporter, right, for the angiogenic potential. So now what we think is that the zebrafish might be also a reporter of hot and cold tumors for innate immunity. Meaning, because we've seen that some tumors are engraft really well. So we inject like 100 fish, for instance, or 200 or whatever. Next day we have beautiful 100 fish injected with tumor cells. And then if we look at 4 days later, we still have around 100 fish. So they're beautiful and engraft really well. But then we also had some other tumors, human tumors also, that we inject, and instead of having 100 in the end of the assay, we only have 20 fish with a tumor. So 80% just simply disappear from the fish, clearance. So we had a huge clearance. And this observation was, well, maybe the first hypothesis was maybe, well, the fish, it's a fish, it's not a mammal, tumor cells are just dying. But it wasn't the case. So we saw that chemotherapy could increase the number of, or could increase engraftment. So the number of fish with tumors in the end of the assay. So this was very paradoxical.
Yeah, I was going to say it's backwards from what you would think.
Yes, exactly. But if you think, and then generating a new hypothesis, which was that, well, maybe chemo is immune suppressing the fish, and that's why you have more tumors now. So we started digging in, and that was the case. So macrophages and neutrophils were actually actively clearing these tumors. But then we did some really cool experiments. I don't know if we have time, but so it generated this hypothesis. Maybe there are tumors that can block this innate immunity and generate an immune suppressive environment. And so that's why they engraft so well. And other tumors are so reactive that they are cleared. So then we mixed both tumors painted ones in one color and others in another. And we just, okay, can these ones protect those ones from being rejected? And then we saw the yes. So there are tumors that are able to suppress this reaction and protect the others from being cleared. And then we went to see that which are the cells that are involved in this. So these are macrophage and neutrophils. And so in terms of application for medicine, we think that first this might help us help in the future maybe select patients for immunotherapy. So not all patients respond well to immunotherapy. And it could be just tumors are not immunogenic enough, or they don't have a lot of mutations, or maybe they generate such a suppressive microenvironment that then even if you boost, T cells, you still have an innate, very suppressive environment. So the idea is that maybe the ones that get rejected in the fish are the ones that will be good candidates for immunotherapy. And then another application is screens. We can do chemical screens, of course.
So you've just expanded the toolkit through this new, almost nuance in a way that you've discovered about the way tumors are behaving. That is fascinating. We're on our last minute because that's how fast this goes when we're having fun. I wanted to turn to another question is that I understand that you have a personal passion for supporting women in science and STEM education. And I think I heard that you've had at times, maybe even now, all of the members of your lab are women. So I'd love if you could talk to us about this a bit more and sort of how you're mentoring this group of women in your lab and advancing.
Yeah, we are almost 100% girls. Now I have one boy at the lab. We have one boy. I must say this is not on purpose. It's just, I don't know, it happens because I think in Portugal we have a lot of girls wanting to do science. So I don't think we need to push. The thing I feel when people ask me about women in STEM is really, I think the difficult part is to balance with family, the family balance, because science, you need really a lot of dedication, and so you need help at home to balance both things. And in that sense, I think I have so we had a lot of babies this year in the lab.
That's wonderful, though, that you provide a supportive environment where women feel.
Yeah, I try. I think
balance.
I think I try to
you walk that line. So here goes our
that.
Goodness me, that was 17 minutes, just like that. Happens so fast every time. This was one of the conversations where I wish we could go on for a lot longer because this was so interesting and fresh with your new publication. And again, congratulations to you and all your teammates on that.
Thank you so much.
Thank you, Rita, for taking the time at the end of your day to join us. And we look forward to following your progress.
Okay, thank you so much.
Well, thanks everyone for tuning in. We look forward to seeing you next time. Until then, be safe. Take care.